All right, today we're going to dive deep. I mean, really deep inside our own bodies, right down to the cellular level. We're going to tackle a question that's totally fundamental to life itself. How in the world do our cells actually handle water?
I mean, really think about that for a second. You have trillions of cells, and every single one needs just the right amount of water to do its job. If they get too much, they could literally burst. Too little, they shrivel up and die. So how does your body manage this insane balancing act? For the longest time, the thinking was that water just kind of seeped through the cell walls. But as it turns out, the real story is so much cooler, so much more elegant, and unbelievably fast. So before we can really appreciate the solution, we first have to get a handle on the problem itself. And it all kicks off with a process you probably remember from high school biology class. And that process is osmosis. You know, at the end of the day, it's just nature trying to balance things out. Water always wants to move from a place where there's a lot of it to a place where there's less of it, meaning there's more other stuff like salt or sugar dissolved in it. Think of water as this little peacekeeper. It's constantly moving across membranes to dilute the side that has more stuff, more solutes, until the concentration is basically equal on both sides. This is happening all over your body, all the time. But, and this is a big but, there's a catch. While water can kind of slowly diffuse across the cell membrane, for some really important parts of our body, slowly just doesn't cut it. Not even close. Your kidneys, for instance, process about 180 liters of fluid a day. That's insane. And your red blood cells? They have to change shape and volume in a split second as they squeeze through tiny capillaries. A simple slow leak was never, ever going to be fast enough. So the cell needed a better way. It needed a superhighway for water. And that right there brings us to the hero of our story, an incredible little protein that acts as the cell's own specialized high-speed plumbing system. This is what solved the cell's water problem for good. And you want to know the best part? It was discovered completely by accident. The Nobel laureate Peter Agre was studying Rh blood types when his team kept finding this mystery protein. They honestly thought it was just some contamination, some gunk in their samples. But nope, it turned out to be the very first water channel ever identified, which he called CHIP28. It's just a classic story of a happy scientific accident. So what exactly are these things? Well, aquaporins are proteins that basically fold themselves into a perfect little tube, or a channel, that goes right through the cell membrane. You can just think of them as the dedicated plumbing system for our cells, letting water rush in and out in a super organized and incredibly fast way. And when I say fast, I mean mind-bogglingly fast. A single aquaporin channel can shuttle up to 1 billion water molecules across the membrane per second. Let that sink in. 1 billion. It's just a staggering speed, and it's all possible because of its brilliant, elegant design. But hey, this isn't just a simple pipe. The structure of an aquaporin is a true masterpiece of molecular engineering. It is designed for both unbelievable speed and pinpoint precision. It all really starts with its shape. The channel itself is formed into this really distinct hourglass. You know, it's wider at the ends, but super narrow in the middle. And that shape is absolutely crucial, because that narrowest point is just wide enough for one single water molecule to squeeze through, but nothing big. So the water molecules have to line up and pass through in single file. And right there at that narrowest point, the channel uses these precisely placed chemical gatekeepers. And these little guys do 2 amazing things. First, they make sure only water molecules can fit. But second, and this is the really clever part, they grab each water molecule and give it a quick spin, flipping it over. This little twist actually breaks up any chains of water molecules and prevents other charged ions, like protons, from sneaking through. It's like a gate that only water has the perfect key for. Okay, so now we know how water zips in and out of our cells so efficiently. But let's talk about what happens when that perfect balance gets all thrown out of whack. This is where we get into a concept called tonicity, which is really just a fancy word for how a solution affects a cell's size and shape. Basically, there are 3 scenarios. If you put a cell in a hypotonic solution, that means there are fewer solutes outside, so water rushes into the cell. In a hypertonic solution, it's the exact opposite. More solutes outside, so water rushes out of the cell. And then there's an isotonic solution, where the concentration is perfectly balanced, so there's no net movement of water at all. Everything's happy. Need an easy way to remember that? I've got a great little trick for you. Hypo sounds like hippo, right? So in a hypotonic solution, the cell swells up with water, getting big and round like a hippo.
It's a little silly, I know, but trust me, you will never forget it.
Now, this water movement has some pretty dramatic effects. An animal cell, like one of ours, doesn't have a rigid structure. So if you put it in a hypotonic solution, it'll swell up and can actually burst. But a plant cell is protected by its strong outer wall. It just gets firm and rigid, which is actually healthy for a plant. And in a hypertonic solution, the opposite happens. The animal's cell shrivels up, while the plant cell's inner membrane just kind of pulls away from its protective wall.
So, why does all of this microscopic drama matter to you? Well, because understanding tonicity is absolutely critical in medicine, especially when you're talking about something as common as an IV drip.
So let's imagine a real-world scenario. A patient gets brought into the ER. They're severely dehydrated and their blood pressure is dangerously low. You have to give them fluids, right? But which kind? Making the wrong choice here could be a total disaster.
Okay, let's use our little memory tricks. An isotonic fluid, you can think of this as I'm so perfect, is perfectly balanced so it stays right in the blood vessels. This makes it great for raising blood pressure. A hypotonic fluid, that's our hypo fluid, sends water into the cells to rehydrate them. And a hypertonic fluid, well, that makes the cells skinny by pulling water out, which is incredibly useful for reducing dangerous swelling like in the brain.
So back to our patient. What do? Well, the number one priority is that dangerously low blood pressure. So you'd start them on an isotonic solution to quickly boost the volume in their blood vessels and get them stable. Then, and only then, once their blood pressure is in a safer range, you might switch to a hypotonic solution to rehydrate their actual cells. So from a simple observation to a Nobel Prize winning discovery, Aquaporins have really pulled back the curtain on this elegant system that keeps every single one of us hydrated. And that just leaves us with this huge, fascinating question for the future. Now that we understand the cell's plumbing, what can we do with that knowledge? Could we develop drugs that target aquaporins to treat fluid retention in heart disease, or maybe even reduce brain swelling after a major stroke? The possibilities, well, they're really just beginning to unfold.